Welcome to the Wednesday afternoon lecture series. I'm Gigi Stortz, a principal investigator in NICHD, and it is my great pleasure to introduce today's WALS speaker, Rotem Sorek, on behalf of the Lambda Lunch Interest Group. So Dr. Sorek was a top vote getter for Lambda Lunch for several years in a row. So we are so pleased that he was able to come this week. He's the incumbent of the Rowland and Sylvia Schaefer Career Development Chair, I hope that's right, in the Department of Molecular Genetics at the Weizmann Institute in Israel. Rotem received the bulk of his education at Tel Aviv University, where he earned his undergraduate degree in life sciences, a master's degree in genetics, and a PhD in human genetics. I didn't know that. He followed up with a postdoctoral fellowship at the Lawrence Berkeley National Lab in Berkeley, California, and then returned to Israel, where he went to the Weizmann and has been going up the ranks, moving up the ranks at the Weizmann. So Rotem has had many recognitions and accolades, and included are the German National Academy of Sciences, Leopoldina, the André Deloro Prize for Scientific Research, the Rappaport Prize for Excellence in Biomedical Research, and election to both the American Academy of Microbiology and the EMBO Society. So recognition all around the world. But most importantly, on to Rotem's research, which combines computational genomics, systems biology, metagenomics, high-throughput sequencing, and other modern experimental approaches in microbiology and phage biology. So among the most significant research results are multiple discoveries about the arms race between bacteria and phage. So many people have heard about CRISPR, but that is not the whole story, as you can see here. It's beyond CRISPR. And so exciting findings that have come from the Sorek lab include the discovery of an astonishing number of systems, defense systems beyond CRISPR, the discovery that phages use small molecule communication to coordinate their infection dynamics. Small RNAs and small proteins, which are close to my heart, are involved in the process. And most intriguingly are the parallels between bacterial and eukaryotic defenses. So I think it is some of these topics that are the focus of the talk today. So let me stop there and thank Rotem for coming to visit us in person.
Thank you. Thank you so much. It's a great pleasure and honor for me to give a lecture in this forum, and I thank the Lambda Lunch Forum for the invitation and for hosting me for such an interesting meeting. These last 2 days have been fascinating here. Thank you. I'm going to tell you about our adventure studying the immune system of bacteria and what we learned in the past few years. And bacteria need an immune system because they are so very frequently being infected by phages. Phages are viruses. Like any virus, they locate their host, in this case the bacterium, using a specific receptor on the bacterium surface, and then they will inject their genetic material within the bacterium. They'll replicate within the bacterium. And with the phages that we usually study in the lab, after between half an hour to 2 hours, they will burst open the bacteria, and about 100 phages will emerge from it and search by diffusion for other bacteria to infect. And phages are really super abundant in the environment. They're more abundant than bacteria on Earth, and it was estimated that if you weigh all phages, they weigh together more than the entire humanity itself. So pretty heavy stuff, which we can only see when we look at the electron microscopy. So bacteria are so frequently being infected, and hence they needed to develop and evolve an immune system. And the immune system of bacteria, we know of it for many years now. We have two, or at least what we knew about up to about 4 or 5 years ago, that we have 2 arms of immunity that bacteria have. One is the adaptive immune system of bacteria called CRISPR-Cas. It's a fascinating machinery that allows bacteria to memorize past infection by putting pieces of the phage genome as immune memory within the bacterial genome, and then use these pieces to mitigate future infection by the same phage. We have another arm of immunity that is known for many years now, which is called restriction modification. These are basically restriction enzymes. They would cleave the incoming DNA of the phage in specific motifs, and there's always going to be another protein, usually a methylase, that will modify these same motifs on the microbial genome to prevent autoimmunity, to prevent self-cleavage. But a number of years ago, we started thinking that there might be other defense mechanisms in bacteria that we are not aware of yet. And the reason that we thought that there might be such new defense mechanisms is that, first of all, CRISPR is absent from most bacterial genomes. So most bacteria don't have CRISPR, and then you ask yourself, how do these bacteria survive phages? And also, CRISPR itself was discovered so late in the history of microbiology. It was discovered only 90 years after the first discovery of the phage, and during these 90 years, at least in some of the decades in these 90 years, phages have been the major driving force for studying molecular biology. So discovery of how DNA is organized, how translation goes, genetic code was all discovered based on infection of phages of bacteria. But since E. coli doesn't have a working CRISPR, so CRISPR in E. coli is not working very well, so that's why it was not discovered during all these decades of research. So we thought that there might be other defense mechanisms that E. coli doesn't have, but other bacteria may have, and these, we could discover them. So the reason we were so interested to find new defense mechanisms, new immune systems, is first of all, if we want to understand the evolution, the conflict of bacteria and phages, defense systems are major components of that conflict. So if you want to understand that conflict, we need to understand how bacteria defend themselves and how phages overcome this defense. But from a more practical point of view, it seems to us that in the past, when new immune systems have been discovered and studied, it frequently led to revolutions in biology. So the discovery of restriction enzymes allowed the first genetic engineering. Before that, people didn't know how to do it. Discovery of CRISPR now leads to a revolution in genome engineering. And if you think beyond bacteria, discovery of things like RNAi, which is also an antiviral machine, led to the ability to silence genes in eukaryotes. And the way we work with antibodies, which is another immune system, is also a biotechnologically very rich tool. And the reason, I think, why immune systems are so frequently becoming beneficial as tools is an immune system, by definition, needs to identify a specific molecular moiety, very specifically, to differentiate between self and infecting foreign element, and then do something to that molecule, cleave it or remove it or do something to it. And that's exactly what we need as molecular biologists. We need these precision tools that will allow us to bind, cleave, or remove, or move away, or do something to a specific molecule. So we hope that if we find new defense systems, new immune systems, they might evolve into becoming new molecular tools that would be beneficial for humanity. But the question is, how can we find new immune systems in a systematic manner, given that all these immune systems in the past were discovered just by chance? People stumbled upon them and then realized what they saw later on. So we've been trying to find immune systems through multiple different kinds of methodologies, and we failed in most of them. But the thing that eventually worked for us comes from a very peculiar behavior of defense systems if we look at genomes of bacteria. So genomically speaking, defense systems cluster on microbial genomes in what was defined by Eugene Koonin and Akira Makarova about 10 years ago as defense islands. So these are areas in the microbial genome that are rich in defense systems. So you can see that in the genome of this microorganism, I'm showing you a small piece of that genome, about 50 genes, and you see that genes that code for CRISPR are encoded not far from genes that code for restriction enzymes. This is the restriction subunit, the methylation subunit, and the specificity subunit, and they're encoded not far from another defense gene that is known. And this is something that recurs again and again in many different genomes of bacteria. So we thought that if we can look for genes, or more often gene cassettes, that recurrently occur in different bacterial genomes next to CRISPR and restriction enzymes and are enriched next to these defense systems, we may find new immune systems that were not known yet. And so Shani Doron from my lab, who was a PhD student, very talented student, she analyzed tens of thousands of microbial genomes to look for gene cassettes that are frequently occurring in multiple different genomes next to CRISPR and restriction enzymes. And we actually, to our surprise, we found many dozens of these gene cassettes that were of unknown function back then. So to see whether they indeed function in defense against phages, we developed this experimental platform to test them. So what we do when we suspect that we found a new defense system, we look at all the genomes in the database that have this defense operon, and we select the genomes that are as close phylogenetically as possible to our lab model organisms, Bacillus subtilis and or E. coli. And then we synthesize the DNA and engineer the system into Bacillus subtilis genome or on a plasmid in E. coli, and then we expose these engineered bacteria to a series of phages that, we have an array of phages in the lab that span many different aspects of phage biology, so double-strand DNA phages, single-strand DNA, RNA phages, etc. And we look for cases in which the system we clone into the bacterium protected the bacterium against that phage. And when we started doing these experiments, we were hoping to find one or two new defense systems, which we thought would be super exciting, but we were super, super surprised to find numerous defense systems in microbial genomes. So far, I think my lab described more than 50 different defense systems that are abundant in microbes, and other labs now that did experiments inspired by us found additional many dozens of defense systems. So now we know, since 2018, of about 100 different kinds of immune systems or defense systems that bacteria encode in nature and protect them against phages. And so what you see here, this figure, let me try to explain what you see. This is a figure from our 2018 paper where we described 9 or 10 defense systems. What you see here, for example, is a 2-gene system. This gene is a domain of unknown function. The second gene has a helicase domain, meaning that it manipulates DNA or RNA in some way. And the size of this gene is about the size of Cas9, so it means that it's a pretty large protein. We cloned it from Bacillus cereus into Bacillus subtilis, and we infected the engineered bacterium with 10 different phages from different morphologies. And whenever you see a red color here, it means that the system protected the bacterium against the phage. And this color means that we needed to infect the bacteria by 10 million times more phages or 1 million times more phages in order to start seeing plaques. So this is pretty strong defense. And in the past 4 or 5 years, since the discoveries of these systems, we shifted the gravity of the lab's work from mostly bioinformatics and genetics to much more biochemistry in order to start solving the mechanisms of how these systems work. So this is what very much interests us these days. And so one thing we saw quite early on, which was very surprising for us, is this system, the Toeri system. So this is one of the defense systems we characterize. And it interested us because it has proteins with tier domains. And tier domains are already known to participate in immunity, but in our own immune system, the immune system of humans. So tier domains, it's acronym for toll interleukin receptors. And these are pieces of our toll-like receptors. They participate in our own innate immunity. So this gave us the notion that for some defense systems, there is an ancient evolutionary link between bacteria and human, and more accurately, we have evidence that some immune systems in bacteria gave rise evolutionarily to components of our own immune systems. And we started asking ourselves whether there might be other components of our innate immune system that evolved from bacterial defense mechanisms against phage. And so the answer is yes. I'm going to show you a few examples for such components. So first of all, let's look at this pathway. It's called the Sigus stink pathway. It's an immune pathway in animals that allow animal cells to identify that they have been infected. And basically, this pathway is present in many cells, not necessarily immune cells, but many cells in our body. And it comprises a protein called Sigus, which is the sensor of cytoplasmic double-stranded DNA. And as you know, cytoplasmic or DNA in human cells is not supposed to be in the cytoplasm. It's illegal in the cytoplasm, and that's what we have a nucleus for. And so the DNA in the cytoplasm is perceived as viral infection. And the sensor for that is a protein called Sigus. And the name Sigus comes because the protein, once it senses DNA in the cytoplasm, it produces a unique signaling molecule, which is cyclic GMP-AMP. So these are two nucleotides, GMP and AMP, which are bound one to each other in a cyclicized form. And this signaling molecule accumulates in the cell and binds another protein called STING. STING is an acronym for Stimulator of Interferon Genes. I really envy the acronyms in the human immunology. These are super cool and very informative acronyms, because what STING does when it binds the molecule is it activates a cascade that expresses interferon and other interferon-related genes. So this pathway was described about 10 years ago, 2013, in seminal papers from the Chen's lab. But around the same time, the Mekalanus lab in Harvard found that some bacteria, specifically Vibrio cholerae in this case, can also produce the same molecule, cyclic GMP-AMP. And they characterized the protein, the cyclic GMP-AMP synthase of bacteria. But until quite recently, the biological role of this protein was completely unknown. And the reason I'm showing you this is because when we look at the protein, the bacterial protein, and we look at homologs of that protein, the bacterial Sigus, in many different bacteria, we see that very frequently it's found next to restriction enzymes and CRISPR. So in this strain of Vibrio cholerae, it's found next to a CRISPR. In this other bacterium, it's found next to a restriction enzyme. And in other bacteria, this gene, the homolog of this gene, are found next to other defense systems that we characterized recently. And this characterization was done by Dim Millman from my lab. And moreover, you can see that the Sigus is present as a 4-gene operon. One of the genes in this operon is a phospholipase that I'm going to talk about this gene shortly. And so the reason, the fact that this operon is so frequently being present next to CRISPR and restriction enzymes in microbial genomes led us to think that it might have an antiviral activity. It might be an antiviral defense system that bacteria encode. So as I told you, what we do when we suspect an operon to function as an antiviral system, we take it from its genome of origin, we clone it into E. coli in this case, an E. coli that doesn't encode it, the lab E. coli, and then we infect the E. coli by phages. And this is a typical plaque assay experiment that we do in the lab, very typical experiments done in my lab every day, I guess. What you see here, the lighter parts are lawn of bacteria growing on a Petri dish, on agar plate. And we're putting on this lawn, we're dripping phages, so that we put a few phages here, 10 times more phages, 100 times more phages, 1,000 times more, and more phages in each serial dilution. And the phages, when they touch one bacterium, one phage touches one bacterium on the plate, it will replicate in that bacterium, kill it. 100 phages will emerge from the bacterium, will infect the nearby cells, kill them too. And when we come in the morning, we see this area of clearance, which are the plaques, regions where the phages, a single phage has been, and kill the bacteria. So what you see here is that when the bacteria encodes the system, now we need to infect the bacteria with much more phages in order to start seeing plaques, about 1,000 or 10,000-fold more phages, meaning that the system protects the bacteria against the phage. And this observation that was done by Sarah Melamed and Daniel Cohen in my lab led us to, started an adventure which involved genetics and biochemistry, and eventually we realized how this system works, or at least how the major parts of it works. So we understand now that the bacterial Sigus enzyme is expressed in the cell, constitutively, even before the cell is infected by the phage. Phage infection activates this protein and to produce the cyclic GMP-AMP, but it's not the DNA of the phages that activates. It's something else within the phage infection. The DNA is not a good activator for, or sensing DNA would be futile in the context of a bacterium because the bacterium has DNA in the cytoplasm. There is no nucleus there. So it's something else that activates the protein. We suspect that it's part of the capsid of the phage, or the proteins of the capsid. And when the protein is activated, it produces cyclic GMP-AMP, which then bind the phospholipase that is found in the same operon and activate it. Now it becomes an active phospholipase. And this active phospholipase eats the inner membrane of the cell and kills it. and the cell dies before the phage replication is complete. And I know that for some people it sounds weird that bacteria would kill themselves, being single-celled organisms. Why would bacteria kill themselves as part of their defensive measure against phage? So let me try to explain how this concept works. So basically, very frequently in nature, bacteria live in colonies, either in solid surfaces or in liquid culture. And these colonies are usually isogenic or nearly isogenic, meaning that they have the same genome. They started from one bacterium which replicated, and then you have a small colony of bacteria. And phages don't have a way to be motile. They are drifted by diffusion. So infection of a colony usually starts by diffusion of a single phage into that colony that infects one bacterium. And this phage replicates within that bacterium, and then after a while, phage particles emerge from the bacterium, and they will infect the nearby bacteria, and eventually the entire colony will demise. But if the bacteria have an ability to sense that they have been infected, and then kill themselves, the single cell that is infected would kill itself before the replication cycle is complete, then only one cell is lost, and the colony survives. So basically, genetically speaking, evolution prefers that. And we now understand that most bacteria in nature have at least one defense system that works this way, meaning that will sense the phage infection and then kill the cell. There are multiple principles that govern defense that works this way. One of them is that these systems usually become active relatively late in the infection cycle of the phage, so that defense systems that can overcome the phage without killing the cell could work first, like restriction enzymes and CRISPR should be able to work first. And if they don't work, only then the other defense systems that kill the cell come into action. So we see that very frequently. So around the same time we discovered this Sigus defense system in bacteria, Philip Kranzush and Aaron Whiteley from Harvard Medical School and Dana-Farber Cancer Institute, they found that the bacterial Sigus is just one representation of a larger family of proteins which produced cyclic oligonucleotide. So what they found is that this represents the family tree, the tree of this family of enzymes, the Sigus-like enzymes. Some of them produce cyclic GMP-AMP, and they're found in many different bacteria. But other clades produce other signaling molecules like cyclic UMP-AMP or cyclic UU or cyclic trinucleotides. And Adi Millman from my lab looked whether these families of proteins frequently occur next to known defense systems. So in 64% of the cases, genes that produce, or proteins that produce cyclic GMP-AMP, the genes were present next to known defense systems. But this was also true for other proteins from this family. And this led us to realize that all these proteins participate in defense against phage, and that what we actually see, and this is found in between 13 and 15% of all sequenced bacterial and archaeal genomes, we actually now understand that there is a large diversity of anti-phage signaling systems in microbial genomes. And these systems have a unifying principle of activity. There's always a protein from the Sigus family that recognizes phage infection and then produces a signaling molecule. The signaling molecule could be cyclic GMP-AMP, but could be another kind of signaling molecule. And the signaling molecule binds an effector that kills the cell. The effector could be a phospholipase that degrades the inner membrane, but it could also be a DNA endonuclease that degrades the DNA of the bacterium and the DNA of the phage to kill both of them during infection. It could also be, the effectors could also be transmembrane domain proteins that depolarize the membrane. And we now have a paper coming out showing that some effectors degrade the ATP pool from the cell during infection to abort the replication of the phage. And we call this family of signaling system, we call them in a unifying name called SIBAS, which is the acronym of Cyclic Oligonucleotide-based anti-phage signaling systems. So again, to put the principle of the activity of SIBAS within blue-colored figures instead of the the red colors are before. So the unifying principle is that there is an enzyme that recognizes phage, produces a signaling molecule that binds an effector and activates that, and the effector kills the cell. So I told you that we identified SIBAS, which is found to 13 to 15% of all bacterial genomes. But later on, we found that there are additional families of immune systems that work according to the same principle. So one of them we called Pixar. And this is a study done together with Philip Kranzush, Ben Morehouse from his lab, and Nitzan Tal, a PhD student from my lab. And we found that this family produces cyclic CMP, or cyclic UMP, so mononucleotide, the new kinds of signaling molecules that activate an effector protein. And then we found another family of immune systems that work in the same principle. This is the Toeris system, in which tear domain proteins produce a cyclic ADP ribose isomer, which we will not talk much about here. So altogether, more than 20% of all bacteria in nature, or at least all the bacteria for which we have their genome sequences, have at least one of these defense systems, very abundant defense systems in microbial genomes. Now, since this is such an abundant system, then one asks themselves, how can phages overcome that? I mean, can phages infect bacteria that have this defense system? And apparently they do. So studies from our colleague, as you can see, we have a very deep and wonderful collaboration with Philip Kranzush. In this case, Sam Hobbs from his lab discovered enzymes that some phages bring, which cleave the signaling molecule and inactivate them, thereby escaping defense by phages. And a similar kind of cleaving enzymes were found also in pox viruses that infect us. So pox viruses, some of them will also have an enzyme that cleaves the signaling molecule at cyclic GMP-AMP of humans, something that was also discovered by Philip Kranzush. And in a more recent study, together with Philip, led by Azita Levit from my lab, Erez Zirmiya, Gil Amitai, and Alan Liu from Philip's lab, we found that other anti-defense proteins of phages will bind the signaling molecule very tightly and sequester it, and thereby, again, not allowing the signaling molecule to bind the effector protein and saving the phage from the defense system. All right, so let's go back to the cell-autonomous innate immune system that our own body has. So as I told you, cells in our body have the ability to sense that they have been infected. And I'm not talking about immune cells, not T cells or B cells, but almost any kind of cell in our body has what's called the cell-autonomous innate immune system, a set of sensors that allow the cell to identify that it has been infected, and then do some kind of signaling cascade to activate, first of all, secretion of interferon, and second of all, the production of effectors within the cell that allow the cell to fight against the infection, or at least the cell tries to fight against the infection before it spreads. So, as I told you, some of the sensors are conserved between bacteria and human. The Sigus stink pathway are conserved. But this is not the only thing that is conserved in the cell-autonomous innate immune system of humans. I'm going to show you another story about a protein called viperin, which is an effector protein produced by infected cells after they sense they have been infected, and that has antiviral capacity. So it was known for many years, I think maybe 2 or 3 decades, that the viperin protein, by the way, viperin is another cool acronym. It's virus inhibitory protein, something, something, nice acronym. And it was known that it protects cells from infection. So if you overexpress it, cells are less inclined to be infected by several different families of viruses. But only about 5 years ago, the mechanism of action was discovered by a group, a clever group in Mount Sinai, I think, Almo Lab and Cameron Lab. And what they found is that the viperin is an enzyme that takes the nucleotide CTP and modifies it slightly by removing the 3' OH and installing a double bond instead. And now, instead of a regular nucleotide, this modified nucleotide, which is di-dihydro-CTP, is a poison for the virus replication. And the reason that it forms a poison is because when the virus takes this nucleotide, when the polymerase of the virus takes this nucleotide to polymerize its nascent chain, then usually you take the template strand, you put the nucleotide against the template strand, you remove the 2 phosphates, and the last phosphate, you attach it to the 3' OH of the previous nucleotide. So if the polymerase of the virus has taken the modified nucleotide, there is now no 3' OH, and this forms a chain terminator so that the virus cannot continue to replicate its genome. And this nucleotide is not toxic to human cells, most probably because the polymerase of humans doesn't need to work as fast as the polymerase of the virus and can discriminate between the modified and the right nucleotide. The reason I'm telling you this is because we found homologs of this protein in bacteria in archaea. This was found by Aude Bernheim when she was a postdoc in my lab. She has her own lab, very successful lab in the Pasteur Institute now. And what she found is that there are homologs of the human viperin. These are the red proteins. They're found in bacteria and archaea. And these homologs are very, very frequently found next to CRISPR and restriction enzymes. And so we thought that from this, as you know now, this tells us that this protein has a function, has an antiviral function in bacteria as well. So again, we took a bunch of these proteins, we cloned them into bacteria. In this case, I'm showing you plaques of the phage called T7. So 1 plaque, 10 plaques. So these are huge plaques. T7 has a very short replication cycle, about 17 minutes in 37 degrees. So that's what it forms. The reason it forms a huge plaque after overnight incubation. And what you see is that when we express a bacterial viperin within E. coli cells, we see much smaller plaques and much fewer plaques. So again, this is an antiviral protein in bacteria. And again, this took us into scientific adventure that involves a lot of mass spec and chemistry. And eventually what Aude found is that this family of bacterial enzymes produce a family of antiviral molecules. Some of them are the same like the human molecule, while others are based on a different nucleotide, so either UTP or GTP. In all cases, the OH from the 3' end is dehydrated, and a double bond is installed, and then this poisons the phage replication. So when we take all the bacterial enzymes, the genes that encode these enzymes, and put them on a phylogenetic tree, what we see is that, we see multiple clades that are formed. Some of them tend to produce CTP. Some of them have more tendency to produce the other nucleotides. And when we add to this tree the sequences of all the eukaryotic viperins, like human viperin, mouse viperin, chicken, even viperin from fungi, we see that all of them cluster on a single clade that emerges from the clade that produces the CTP and that is mostly enriched in archaea. So we think we have pretty strong reasons to believe now that there has been a single acquisition event in the very early evolution of eukaryotes in which the early eukaryote acquired one viperin from bacteria, and the evolution of the eukaryotic viperins started based on this acquisition and led to what we see today in humans and in other eukaryotes. Now, one thing that really surprised, again, there are many surprises in this story. So that was another surprise, that bacteria can produce antiviral molecules. We're used to thinking about bacteria as source for antimicrobial molecules, but it's surprising to realize that bacteria produce small molecules to protect themselves against phages. Now, this is a modified nucleotide, and those of you who understand how antivirals work, realize that in many cases, antivirals that we use against viruses that infect us are also based on modified nucleotides. So this molecule in the middle is part of the 3-drug cocktail that people take against HIV, the AZT, if I'm not wrong. And this is a nucleotide that has, instead of the 3' OH, it has 3 nitrogens. This is the drug that people take against herpes viruses, what you put on your lips, this ointment, when you have herpes. This is a modified nucleotide that lacks the bottom part of the ribose. It again works the same way. The virus takes it in for replication, and then it's a chain terminator. So we think, now realizing that bacteria produce antiviral molecules against phages, we are wondering whether these molecules can also work against viruses that infect us, and we're trying to see whether they could be new drugs in the future. Here's another defense system that is a very commonly studied one these days in humans. It's called the Gasterman pyroptotic system. So in animal cells, including in our own cells, there is a system that, or an apparatus called the inflammasome that recognizes infection, mostly by bacteria, but sometimes by viruses as well. And once infection is recognized, a protease called caspase that you probably have heard of, it's becoming activated. And this caspase is, by doing some cleavage event on a protein called Gasterman, which is usually found in the cell in an inactive form. Once it's cleaved by the caspase, it's becoming active, and it makes a giant pore in the cell membrane. And basically, the cell leaks out and dies. At the same time that the cell dies, it secretes interleukins to invite the immune system to the site of infection or the site of death, and that's why this type of death is called pyroptosis, because it's cell death that involves inflammation. Tanita Vine from my lab, she's a postdoc, very talented postdoc in my lab. She discovered homologs of the human Gasterman in bacterial and archaeal genome. And she found that in all cases, these homologs, or almost all cases, these homologs are found next to a protease, either from the caspase family or another kind of protease. So we suspected, and also these are found next to CRISPR, so we suspected that this is another defense system in bacteria. And together with Philip Kranzush and Alex Johnson, a talented postdoc from his lab, they showed that indeed during infection, this is Tanita from my lab, during infection, the caspases are activated. Presumably the caspases sense, or the caspase-like proteases are sensing the phage, activating Gasterman by cleavage. We verified that, and they make giant holes in the membrane that kill the cell. Again, an abortive infection system, a system that kills the cell before the phage is ready, before the phage particles are ripe within the infected cell. Let me show you another peculiar mode of defense that we found in bacteria. This was discovered by Nitzan Tal from my lab, super talented PhD student. She's only in her middle of the PhD, but has been very successful in her discoveries. So what she found is that there are families of enzymes that recognize phage infection. Most probably, they recognize that the phage has inhibited the transcription from the promoters of the bacteria. And once this is sensed, these enzymes take one of the nucleotides, the deoxynucleotides in the cell, which are the building blocks for DNA replication, and basically degrades that nucleotide. So she found two families of enzymes. One is a deoxy-GTPase. These enzymes separate the 3 phosphates from the base and sugar, destroying deoxy-GTP. And another family of enzymes is deoxy-CTP deaminases, which remove the NH from deoxy-CTP and making it into deoxy-UTP. Basically, these enzymes hide one of the deoxynucleotides away from the phage. And so the phage tries to replicate its genome, and without the nucleotide, it gets stuck, and the genome is not replicated. Now, the reason I'm showing you this in the context of conservation between bacteria and human is because we were very proud of ourselves for discovering a new mode, a new strategy to protect against infection, but then we realized that a long time ago, it was already discovered in the human immune system. And this is manifested by a protein called SAMHD1. It's an anti-HIV factor that blocks HIV infection by degrading the deoxy-NTPs into the 3 phosphates and the sugar and base moiety, very similar to the deoxy-GTPase, but it does that for all nucleotides. So we now realize that multiple components of our innate immune systems have parallels in the bacterial immune system. And in some cases, we have very solid proof that the human component has evolved from the bacterial component, so solid phylogenetically based proof. So what we think happened is that, what can explain that is that phages that's very well accepted, that phages have been in the world well before eukaryotes even were here. So phages were infecting bacteria very long time ago, very early on after the bacteria was invented. And so many defense systems have evolved in bacteria before eukaryotes came to be. And these defense systems have been in bacteria for many, hundreds of millions of years. And at some point, the first eukaryote came to be, most probably by fusion of an archaeon and a bacterium. And these archaeon and bacterium used to have defense systems in them. And these defense systems were the basis for the evolution of the cell-autonomous innate immune system that we know today. So this teaches us about the evolution of our own immune system, but it can also be beneficial for understanding immune systems of humans. And what do I mean? We're beginning to find now defense systems that we know exist in humans, but we don't know how they work. So, and we now find them in bacteria. So this is one interesting immune system. It's called ISG15. It's a ubiquitin-like protein that is induced by interferon. It's called ISG because it's an interferon-stimulated gene. And it's induced together with ubiquitin charging enzymes and ubiquitin and deubiquitination enzyme that are specific, and it protects our cells against some viruses. And it has some roles in COVID protection, COVID infection, but we don't know how it works. People in immunology field, they don't know how exactly it works. Now we realize that such a system exists in bacteria. There is an operon of 4 genes, ISG15-like protein, an E1, E2, ubiquitin charging domains, and a deubiquitination domain. And we show that it protects bacteria against phages. So now we're trying to figure out how it works against phages, and we hope that if we solve the mechanism in bacteria, this could translate into understanding of how the human mechanisms work as well. And so we have a bunch of such immune systems or immune mechanisms that are found both in humans and in bacteria, but we don't know yet how they work in humans. We hope to solve this riddle. Now, not all of the defense systems we found in bacteria have homologs in humans. There are some peculiar things that we don't see homologies in humans, and are still very interesting. For example, we found this protein called radar, or this is a system that Kunin and Zhang lab called radar, which comprises 2 very big genes. One of them has an adenosine deaminase domain, the other has an ATPase domain. And together, again, with Philip Kranzush and Brianna Duncan-Lowe from his lab, they show that these proteins form a giant complex in the cell. This giant complex is built from 12 monomers of this. They form like this kind of a soccer ball, and the active sites of the adenosine deaminase are on the faces of the soccer ball. And this protein forms a heptamer. And each heptamer sits on one face of the soccer of this dodecamer to form a giant complex that is 4 times as heavy as a ribosome, so a very big complex. And what we showed is that this complex converts massively ATP to inosine triphosphate during infection. We don't know yet how it protects against phage, but this conversion mediates protection against phage. We're still studying that. The complex is something like, we haven't seen anything like that. Very peculiar why a cell needs such a giant complex in order to mediate this biochemical activity. Another thing that is very interesting to us, we found, is something called Retrons. This is a very peculiar entity in bacteria. It's comprising a non-coding RNA and a reverse transcriptase that reverse transcribes parts of the non-coding RNA into cDNA, leaving the cDNA covalently bound to the non-coding RNA. So this is a chimera between RNA and DNA that was known in bacteria for many years. It's found in more than 10% of all bacteria. And the function of this was not known. We found that it participates in defense against phages. And we found that one of these, it's called a retron, one of these retrons is a guardian of another defense system. So there is a defense system, or there is a complex called RecBCD in the cell that is involved in recombination or repair, but it also cleaves linear DNA. And you know that when the phage injects its DNA into the cell, it comes in a linear form, so the RecBCD eats this DNA very rapidly. And for this reason, many phages are also bringing proteins to inhibit RecBCD. We found that one of these retrons guards the RecBCD. And when the phage inhibits that, the retrons come into action, and then, together with another protein, kills the cell. So it's a guardian of another defense system, and if this other defense system becomes inhibited by phage, this guardian kills the cell. Very peculiar. We don't yet understand why you need a chimera between DNA and RNA to perform that function. Now, of course, there are many open questions. This happened very fast, these discoveries, and we still don't understand much. So there's much that we don't understand. So, for example, for many systems, we know what happens after they recognize the phage. You know, in some cases, they produce a signaling molecule and kill the cell. In other cases, you know, they do something else. But we don't know what is being recognized, how they recognize phage infection. And Avigail Stocker-Avichail from my lab, she recently did a very large screening experiment where she took all the defense systems that we have, about 50 defense systems we have in our lab, and each defense system, she infected the cell that encodes that defense system with a phage that is normally blocked by the system, and she searched for phage mutants that can overcome the defense system. Then she sequenced the phage genome, the mutant genome, to see what got mutated. And in many cases, the protein the gene that got mutated is the gene that gets recognized by the defense system. And she realized that defense systems frequently identify either components of the core replication machinery of the phage. These are three proteins, usually the helicase primase, the polymerase, and a single-strand binding protein that form the replication machinery. This is a very relatively conserved entity within the phage biology that's very hard for the phage to change. So many systems recognize one of these three proteins. Other systems recognize structural components of the phage. Could be, you know, the tail 2 protein or the capsid or something else in the structure. And some other systems recognize that the phage is taking over the cell. You know, some of them recognize that another defense system gets inhibited, and some others recognize that transcription becomes inhibited in infected cells. And this becomes something like a unifying principle in how defense systems recognize infection by phage. Another open question is, what are those defense islands? So I showed you that we identified multiple defense systems based on their presence frequently next to known defense systems, and this defense island was very fruitful for us for discovery of new systems. But the question, what are defense islands, bothered us for a number of years. And Dina Hochhauser, a PhD student in my lab, she found that defense systems are carried usually by mobile genetic elements. So this is a study in E. coli, and she showed that different strains of E. coli have different sets of defense systems that are carried on dedicated mobile genetic elements. So what you see here is E. coli K12. This is the lab strain that lacks most of the defense systems that we found. And these are different strains of E. coli. You see that in each of these strains, an island, a mobile island, bringing each time a different sets of defense systems have integrated in the same place. And so we now understand that about 90, if you look at, you know, we looked at about 1,000 E. coli genomes, and we see variability in defense systems in these genomes. Most of the defense systems, 90% of these systems are carried by mobile genetic elements. So here, this is a conjugative element that brings different kinds of defense systems, again, into the same position in the E. coli genome, but in each strain, it's a different set of defense systems forming these defense islands. And this panned out into something that we didn't expect in terms of the distribution of defense systems in microbial genomes. We see that they are very sporadically present in microbes. So what you see here, each row here is a strain of E. coli, and each column is a defense system. So this is a restriction enzyme type 1, type 2, type 3, type 4, other defense system that we discovered, CRISPR. Whenever you see the colored box mean that this strain has the system, and you see that these systems sporadically occur in strains. The first strain has this disarm system, and Durantia system, and Zoria system, and the third strain, has a type 4 restriction enzyme, but not type 1 restriction enzyme. It has a Gabbage system, etc. And so these systems are sporadically occurring in different strains of the same species, and we call that the pan-immune system of bacteria. We think that a single bacterium cannot encode all the defense systems at once because the tiny toxicity that emerges as autoimmunity from each of the systems accumulates to too much of toxicity. And on the other hand, other bacteria that encode these systems in a nearby environment could contribute that through horizontal gene transfer, and these systems could be acquired by strains in nature if they're needed. And so I want to end by showing this poster. We were asked to put what we know on defense systems of bacteria on a single poster. Obviously, it's too small for you to see. CRISPR-Cas is here. Restriction enzymes are here. And what fascinates me about this poster, which is already obsolete, we published that in the beginning of the year, last year, and now many additional systems and mechanisms were discovered since then. But what really fascinates me is that many of the mechanisms that are described here are only the product of studies of our lab and many other labs in the very recently. So, much of the immune system of bacteria we just didn't know, and now it's like, lifting or digging a giant excavation site and realizing there has been a city below. And moreover, what you see here in a tiny font are just a list of names. These are the names here of defense systems that we know they protect against phage. We don't know yet how. So very large, grounds for having, making fun biology and future discoveries. And I want to end with this by thanking my very, my lab, which are a very talented group of people, very glad to help with them, to work with them. And also our dear collaborators, Philip Kranzlush and many members of his lab. We've been having a wonderful collaboration over a very short time period, and most of this collaboration was done under COVID, so we haven't met until recently. We published multiple papers together, but only met once, not in Zoom, that I mean. And these are our funders, and thank you very much for listening.
Well, of course, anyone here is free to use the mics, and as you're making your way over there, I do have a couple questions from online. This one comes rather early in the talk. Does expression of viral capsid proteins lead to cell death via the CGAS system?
It's a great question. So in some cases, in some defense systems, expression of the structural components of the phage is directly leading to toxicity, leads to cell death. In the case of SIBAS, we couldn't find another system called Pixar, where we also found mutation in the capsid. When we express the capsid protein, there is no toxicity. So we think that although when you mutate the capsid protein, the defense is gone. So we think that either the larger context of the phage that's being assembled within the bacterium is the trigger for the SIBAS activation or the Pixar activation, or you need multiple signals, both the capsid and something else that we don't know of yet. So we don't see activation just by expressing the capsid for this specific case.
You may have touched on this right at the end, and I may have just missed it. Do you find the bacteria stealing pieces of the phage to use as, making abortive pieces of capsid that destroy the ability to assemble it, or making subunits that don't work, or other sorts of things?
We don't find evidence that bacteria, that the defense systems that are within these defense islands, we didn't yet find evidence that they comprise ancient pieces of phages. It doesn't mean that this is not a strategy of bacteria. It could be that some bacteria protect themselves against the phages by this, but maybe these proteins are not traveling on defense islands like these systems.
Yeah, let me follow up with a double question about defense islands. No question is an island, I guess. Are defense islands transcriptionally organized similar to their temporal response to phage? And do defense islands defend against other threats like competitor biofilms, protein aggregates, or prions?
Right. So based on the organization of the genes in defense islands, we don't think that there is or activation by gene expression. So what we see frequently is that the defense systems we discover are expressed in the cell and are found as proteins even before the phage infects. The reason we think that this is essential is because some phages are very quickly killing the cell. So, some phages would degrade the entire genome of the infected cell after 4 minutes from the onset of infection. This is something that T4 does. And so this is not enough time to activate expression and then defend. So usually the activation of the systems is done in the protein level. There are evidence that you can elevate the expression of some defense systems using some external signals. Some of them are involved in the density of the culture, and some of them are not known yet. So that's the answer for the first question. And the second question, do defense islands protect against other challenges?
Competitors like
Other
biofilms,
competitors.
protein
Yeah.
aggregates, prions.
Not that I know of. I mean, we've been studying the defense systems in an isolated manner. Basically, what we've been doing is we never took a defense island and installed it in E. coli. We took one defense system, put it in E. coli, and studied what it does. So we don't know if these islands do other stuff.
So I noticed earlier in your talk when you were giving us specific mechanisms, defense mechanisms, that the names of the mechanisms were acronyms based on what they do.
Right.
But at the end you gave us a list of 50 names that sounded a lot more imaginative. Where are those names coming from?
Right. Thank you for this question. So yeah, we started by calling the defense system with, the first system we ever discovered was called BREX, which is bacteriophage exclusion. The second one was DISARM, which is defense island system associated with restriction and modification. But then we ran out of acronyms, basically. So we started calling them by names of protective gods from different mythologies. So this Gabia is a protective Lithuanian god. Druantia is a Celtic god of the forest. Wadjet is an Egyptian goddess. So that's the names.
On a more serious note, since a lot of the mechanisms kill bacteria, is there an obvious way, and I'm sure you've thought about this, to use those mechanisms as to activate them as antibiotics?
Yeah, this is a great question. I'm not sure how useful would this be because the systems are so sporadically present in bacteria. So, the SIBAS system, which is present in about 13% or 15% of the genomes, you can imagine that if you give them the signal in high enough concentration, the cyclic GMP-AMP, some of it will leak into the cell and will force the cell to die. But some of them use cyclic UMP, some of them use cyclic trinucleotide. So you'd be able to only kill a small number of them. And since it's not that you'll be able to kill all the E. colis, because the systems are sporadically, you don't see that. The systems are sporadically present within different strains of E. coli express different systems. So you'd be able to kill just specific strains of the bacterium, and it will not be profitable for a drug company to be so specific, I imagine.
Yes, there are. A colleague from yours and probably in Switzerland for the Wednesday evening lecture series we have there, Vels. Luke Loof. Great talk, Rotem. Why are restriction enzymes so prevalent compared to other innate defense systems?
Well, restriction enzymes give a very good ability for the cell to protect against the phage without killing itself. So that's a good reason to keep them there. Some phages bring anti-restriction. That's something very common actually in phages. proteins that inhibit restriction enzymes, and so in cases where your restriction enzymes are inhibited, I guess the cell would want to activate the system that leads to death.
It's very fascinating. I worked 50 years ago on the subject of when T4 phage infects E. coli, the very first thing what happens, at least I don't know now which strain was that E. coli strain, but E. coli, it immediately inactivates the RNA polymerase for transcription of the E. coli DNA because it ADP ribosolates the sigma factor. And only this ADP ribosolated RNA polymerase, the sigma factor, it can transcribe the T4 DNA. And that's how then the whole process proceeds down the road. So that is, I think, long back discovered, very first thing that the modification of a very important sigma factor which is involved in the initiation of transcription of RNA DNA.
Yeah, yeah.
Do you have another question?
So,
Do you have another specific question?
No,
okay. So maybe one last question online. We're just past the hour. Given the homology between bacterial and eukaryote immunity, is there any evidence for homology between anti-defense genes of phages and viruses attacking eukaryotic cells?
It's a great question. We haven't found, so far we didn't discover too many phage proteins that inhibit bacterial defenses, maybe 4 or 5 such families of phage proteins. And we haven't found homologies to, proteins in viruses that infect humans. Sometimes there is analogy. So as I told you, there are enzymes that phages bring that cleave the signaling molecules of the defense system. And there are enzymes that pox viruses bring that cleave the same signaling molecule in human, but these are not the same enzyme family, so they evolved separately in both lineages as far as we understand, so there's no structural or sequence homology there. I imagine there might be, but we haven't found them yet.
One more, if you don't mind?
Sure.
Okay, last one, I promise. Do you see any role for modified nucleotides made in bacteria in a microbiome context being taken up by the surrounding eukaryotic cells? Are these absolute conversions of the entire pool inside the cell?
We haven't looked into that. It's a good question, but we haven't explored it.
Okay. My lips are sealed.
On that note, on behalf of Lambda Lunch, I want to thank you for such a fascinating talk and the audience for such a great discussion. I want to invite everybody here live and present in the audience to a reception with cookies and coffee sponsored by Lambda Lunch. They're real cookies and real coffee. So please join us out there to continue the discussion. So thank you very much.
Thank you very much. Thank you.